managed to acquire the unusual clots that have been bandied around on the internet. It's diseased tissue, and it's proteinaceous and primarily misfolded fibrin. What I can tell you is that this proteinaceous form is, like I say, it's misfolded, amyloidogenic fibrin, okay? And this is occurring in the blood of people. I would argue a self-catalyzing, reinforcing phenomenon once the initial seeding event has happened. And this is, again, this overlaps with what we understand with amyloidogenic prion-like disorders. There's a seeding, and then there's an amyloidogenic cascade, which leads to the manifestation of the disease as we understand it. And it's not just immune dysregulation at this point. The peptide itself, as we'll see, is causing this buildup of toxic peptides in the body itself. That you're dealing with any systemic amyloid type disorder is considered along the sort of scale of disorders that you can turn up to a hospital with as being one of the more serious conditions that you can develop. It can manifest in a number of different ways. Okay, cancer is one, neurodegenerative is another. In this instance, if your circulatory system is filling up with this undigestible amyloidogenic peptide, you're going to develop all manner of cardiovascular and thrombotic type tissues or disease. All of it fits under the umbrella of proteinopathy. Proteinopathy means misfolded peptides, and there are hundreds of identified peptides now that are implicated in any number of diseases. In fact, mass spec analyses are not as good at looking for this amyloid stacking as the Raman spectroscopy is. Raman spectroscopy is in fact turning out to be one of the best methods that we have. Problem is that a Raman scope will cost you $1 million, and very, very few people have access to it. Suddenly, there looks like there's an infective peptide. If this sample that I've been running has been out of the body for 3 months, even though it's been out of the body 3 months, once it comes into contact with fresh plasma, is able to initiate the misfolding and aggregation into a amyloidogenic form, such that you're seeing an increase in fluorescence. This is a big concern. I can take people's blood, I can process it histologically, I can look at it under a microscope, and I can tell you, I can give you an assessment of how much amyloid you're carrying per sample of blood, when we potentially can look for pathological signatures and say, look, there's something we need to be treating, and there are methods to at least try to mitigate amyloid burden, especially if it's in an easier to reach compartment of the body as the blood. Nearly, I would say 90% of the people that have sent me blood, when I've written back to them with their results and said, look, here you go, you've got this, I'll send you a full report on all the images that I take, and often I'll send people I'll video record myself working through the slide and just giving my commentary, send that back as well. And that gives people hope and something to go to their health practitioner with and say, this is where we need to be working. And with that, you can start working towards trying to regain your health. Are you going to get back to normal? I don't know. I'm not saying that. I'm not a doctor.
Hello everyone, welcome back to Health Alliance Australia's podcast. My name is Jeannie Rose Angewartha, and I'm here with neuroscientist and distinguished researcher, Dr. Kevin McCairn. Today, we are doing an important follow-up to our podcast recorded last year on biowarfare prions. Dr. McCairn and other researchers have now confirmed through laboratory analysis that we have a global amyloidogenic health crisis. These amyloidogenic prion-triggering epitopes and peptides are also seeded in the environment as well as in living organisms. They are highly resistant with the potential to cause multi-organ dysfunction, neurodegeneration, cancer, heart disease, sudden death, strokes, etc., etc. Dr. McCairn has found a very novel kill mechanism. This research is cutting edge. The latest laboratory findings should be the biggest news story out there right now. It affects every single person in the world. Due to censorship, the bulk of the podcast will be on Rumble, BitChute, and Vimeo. Watch to the end. We will be discussing the detection of amyloid burden through blood samples that you can send to Dr. McCairn's lab from anywhere in the world. If you have any concerns or would like to simply submit a sample for research purposes. There will also be a link if you want to contribute financially to this important work, which is completely community funded. Welcome, Dr. McCairn. Thank you for coming today. For those who don't know you, could you please do a short introduction and then we can get right into the clinical findings and implications?
Sure. So I think you did a pretty good job of sort of the more recent, but my history as it goes back into past decades was, as you're saying, neuroscience, systems neuroscience, disorders of corticobasal ganglia systems. I developed the first primate model of Tourette syndrome, but I also worked on Parkinson's neurodegenerative disorders, surgical interventions thereof around a technique called deep brain stimulation. And basically I got dragged into COVID very early on. I had a very severe case from what was basically Asia's biggest super spreading event that came out of Korea. It was touch and go with me as it sort of took a hold. And as that was sort of unfold, I was going through that, word began to come out of events going down in Wuhan. And, you know, I'm familiar with most viruses, just having, you know, you run into norovirus, influenzas, etc. as you traverse through this life. And this was something standout that I ran into, and as I was watching everything unfold, and I spoke up very early about, because I had predisposing factors anyway. I was sort of semi-medically retired because of a TBI, but my interaction with COVID was what would be defined now as neuro-COVID. It was, it was just a tough few weeks, and then there was a very long recovery process afterwards. And as people were talking about possibility of leaks from Wuhan, I spoke up publicly and said, you want to pay attention to what this is. It's, it did a number on me, and it's obviously done a number on many other people. The issue you have is that, well, since we spoke last year, there's been a sort of sea change in how the public, not the public, the institutes have accepted the fact that there is a synthetic origin to COVID. But the whole mess that emerged from that literally left tens of millions dead, and probably orders of magnitude injured and incapacitated. But in the preceding time between when we last spoke and now, I've managed to acquire the unusual clots that have been bandied around on the internet and diseases, and so I want to say it's almost coming up to 3 years since these samples were first sort of being spoken about by morticians who were finding them in bodies that they were having to prepare for burial. And it's a shame because as a consequence, there's been a whole series of absurdities built up around what they actually are versus what you can see with the current cutting edge technologies required to look at these well, it's disease tissue and it's proteinaceous and primarily misfolded fibrin. Okay, and so I've received them, and the people who were holding onto them or who were trying to do analysis on them, they didn't have the facilities to just even do histological sectioning, etc.
Yeah.
And I do have those facilities and access to a whole bunch more that can allow really thorough probing of what this material might actually be. Now, and so what did I find once I received these samples? So first of all, I did receive these samples, and you know what, I'll just go and grab one just so we can just talk about it. Okay, so I don't know how close, but there is one right there. I have 13 different samples from different individuals who have been unfortunately had to have been dressed for burial, and the age range is wide from 30s to old age. And what I can say is that one, prior to me receiving them, I had no idea how much validity there was in what people were saying online. And I'm going to dismiss the more lurid stuff, but is there something that constitutes a phenomenon that people, although they're talking about it to some extent, that if it gets handed to someone like myself, I can look at it and say, okay, here's what the properties it has. This is what happens when we begin the analytic process. And
yes,
this is a sample with a little bit of alcohol in there and then under argon gas. And what I've been able to do now is say I'm confident now that yes, they're real. I've had plenty of experience now in just handling them, cutting them, using them for different experimental designs, etc., and trying to find out exactly what the properties are. Now, before we sort of get lost in details, what I can tell you is that this proteinaceous form is, like I say, it's misfolded, amyloidogenic fibrin, okay? And this is occurring in the blood of people. Now, this is, you can consider this a separate disease pathway than the one we were talking about last year, which is the potential concern about amyloidogenic attack epitopes on the spike protein. And in this instance, we were talking about the Hamstrom preprint, which showed that it had a high specificity for the prion protein and its conversion to the scrapie form. This is different to that, but there is a commonality in the disease mechanism that between scrapie prion, misfolded peptides in Parkinson's and Alzheimer's, and what we're looking at here. And what we have to try and find out now is what is the causal mechanism for the formation of these misfolded fibrin in people's.
I'm hearing what you're saying is that, correct me if I'm wrong, that last year we were talking about the spike protein causing a disease state. Now what you're discussing is the spike protein infects the body, and then the body is producing more spike proteins or whatever this thing is that is causing these clots. And therefore, I think I heard somewhere you saying that eliminating the spike protein itself from the body once the process has been seeded is of no effect, or may not be helpful.
It might not be enough. So
Yeah.
just let's think about what we're looking at here, which is I've took the gloves off, so I don't want to handle them too much. But I'm essentially receiving samples in the tens of grams in weight, okay? And people's exposure to spike protein is going to be in the picogram level. So this phenomenon is, I would argue, a self-catalyzing, reinforcing phenomenon once the initial seeding event has happened. And this is, again, this overlaps with what we understand with amyloidogenic prion-like disorders. There's a seeding, and then there's a amyloidogenic cascade, which leads to the manifestation of the disease as we understand it. And so, so at this point, what I've been doing, well, actually, let me sort of take a step back. So prior to receiving these samples, which have come from cadavers, what I was trying to do was to take the spike protein itself, and it's possible to buy spike protein. It's called, technically, it's a recombinant peptide, which is it's kind of similar to how the gene transfection worked with respect to vaccination campaigns. But what they do is they'll have an expression system, usually bacterial, or there's a common cell line called HEC cells, and you can force those expression systems to make you a whole bunch of spike protein. And what I've been doing is, well, for a year, so I was setting up the lab, and at the same time trying to work with a technique called RT-QuIC, which stands for real-time quaking-induced conversion, which basically just means you have a well plate, 96 well, where you put samples in, and then you shake it violently for 30 seconds, and you look to see if there's any buildup in fluorescence where the dyes that we use to detect amyloid aggregation would bind to any products that are forming it in your tray, and that gives you some idea as to the amyloidogenic prion catalyzing potential within those peptides. Now, I was able to, well, let's just say using the recombinant peptide approach, the results were ambiguous, although I got some positive hits. The hits were weak and not a, sorry, I'm just getting beeps and alerts in my corner. Let me just see if I can mute that real quick. So where was I? Yeah, so in terms of recombinant peptides, trying to, well, to recapitulate what Hammerstrom was talking about, I was having very little success, and very little success with an animal model platform in which to test. And it's still going. Hang on, let me just. Close that. So I was having very little success in that regard, and it was frustrating because you're well, one, we're not a funded government institute, right? We're trying to do these experiments off a very limited budget, and essentially my time and commitment and well, ever expanding strings to my bow as I try to develop techniques to examine what it is that we're looking at and seeing if we can it's just Jesus, I'm getting so many alerts. Stop, what's going on? Skype.
You're such a busy person. Kevin.
Yeah, it's just the wrong time. Let me just switch my phone off. Power off. Thank you. Dropped my sample on the floor.
But you're working from a hypothesis that the spike protein produced these.
Yeah, so there's an amyloidogenic potential, right?
Yeah.
And so that amyloidogenic potential is there with the virus, and most likely there with people who receive the vaccine. In that space, we have to, because the institutes have been so inherently dishonest, we have to find what are reproducible phenomenon and reproducible enough to allow testing of countermeasures. That what we're trying to do is basically open source, and
Find the cure.
I wouldn't, cure is a big word, but yeah, treatment, because you're dealing with a complex disease process. There are no cures for amyloid disease states. And so we've been looking towards trying to find, just open source, non-patent interventions that people could take, and if they work, great. If they don't, then back to the drawing board. But and so let's sort of get back to the original question, which was, last year where we were talking about hypotheticals. At this point, we're now being able to look at concrete, not concrete in the literal sense, but actual tissue that has been described by individuals as being unusual in its appearance in terms of timing. In the claims are that the people who are first seeing these, I don't want to say first seeing them, but are seeing them where they say they weren't seeing them a few years before, now trying to figure out what to do with these samples as it makes their job more difficult, because they can't dress the body as they normally would, because the vascular system is blocked with these types of proteinaceous obstructions. And so it's I want to say fortune has shined on us, because it's just so unfortunate, the source of where they've come from. But now we basically have a huge
forensic evidence almost.
Yeah, well, yes, that's something we can get to later on in the discussion, because this is it's important factor that we need to think about. But in terms of having a source of disease tissue, right, where I've spent a huge amount, not only of time, but in financially trying to, like, these recombinant peptides are not cheap, right? It's $1,000 per 100 micrograms, right? And that lasts a couple of experimental sessions. And so where we were, or I was struggling to sort of get something reproducible, now we're in a position to be able to say, okay, we have this tissue. What do we see from the macro gross anatomy to the microanatomy to the molecular? And unfortunately, in terms of public health, this all the evidence points to this being amyloidogenic in nature, and I can walk you through the slides and the analysis that we've done that shows what it is that we're dealing with. And I don't know if you want to.
Have you actually managed to test any of these clots or whatever, calamari clots that have come from the living? Like, you know how it's in a blood test.
No.
Has anyone managed to send you that? Because I know that, you know, you see it anecdotally, various people posting that they've come up. But
Okay.
obviously we don't know what they are because they haven't been tested yet, if they're exactly the same as what you've
Right.
seen.
So let's sort of set the record straight with white fibrous clots. White fibrous clots are part of the landscape of phenomena that is occurs in the cath lab and surgery when people turn up at a hospital and are suffering from stroke, blocked arteries, blocked right, and so the apparently runs to about 15%. Now, what I, you know, there's been a lot of discussion about, well, why aren't people seeing these or reporting these in the living? And I think the answer is that post-COVID and the vaccines, just the system has absorbed people turning up with these clots and are basically they're probably just putting it down to, this is the new world in which we live in, which is, we know that COVID, and they admit to vaccines having the primary adverse event profile is cardiovascular and thrombotic events. Okay, so I think what's happening is those cath labs, etc., are seeing these phenomenon. It's just put down as yeah, it's the world we live in now. It's COVID related. They certainly aren't going to be looking at vaccines because it's just not worth the trouble for them. And they'll do the treatment. Probably the clot goes into clinical waste. And unless there's money and an active research program to be looking at these clots, then there's no reason for them to start dissecting them and trying to understand the actual nature of them, right?
Yeah.
And that's unfortunate, and it just rides on the fact that white fibrous clots have always been with us, okay? And
But you're saying this thing is quite novel, what you're seeing within these deceased bodies.
no, let me be very clear here, right? I'm a neuroscientist. I worked around misfolded peptides that occurred within the central nervous system. I have zero experience in cath labs. The closest I get to working with cardiovascular systems is termination experiments where I have to extract tissue, and that involves perfusion of, where I work with primates. You essentially, it's transcardiac perfusion is the technical name for the procedure. You open the chest like you would in an open heart surgery, cannula inserted into the heart, and then I'll pump out the blood and pump in formalin to preserve the tissue ready for histological processing. And in that process, I see a whole bunch, the blood will clot as it comes out of the body. They're typically these red jelly-like clots. I don't ever remember seeing white fibrous ones. And white fibrous clots, or generally clots, when people go to the cath lab, I think generally there's something's been building up over years, if we think prior to COVID, that is, and eventually some sort of catastrophic state is reached. They go for a treatment, emergency treatment, and you know, such. That's the framework in which we were always working. And I think that because anyone who's sort of responsible for the surgical procedures is going to look at what comes out after they've done the thrombectomy, which is basically mechanical extraction of these clots, and just put it down to, yeah, it's that form versus this form. And again, it comes down to, is there a active research program looking into these clots to say these are different to clots that were retrieved previously to COVID? Now, I would very much like to get my hands on a pre-2020 series of white clots to see how much they overlap with what I'm looking at here. I think what's unusual about these ones is just the size and scope of them, and the fact that if the morticians, well, we'll just give them that they are telling the truth in this instance. But it's also a possibility that just because of the the unusual societal dynamics that took place as they shut people down and forced mandates, etc., that maybe, you know, what was just ignored previously suddenly gets attention now from morticians. That's something that we have to sort of hold in our minds as well. And so all we can do at this point is forensically examine this tissue, like I said, from the macro down to the micro anatomy to the molecular, and then see if there's anything that we can tie back to the causal agent. And like I say, just to forewarn people, when you're dealing with something that potentially where you had a catalyst in the picogram range, and suddenly you're dealing with masses of tissue that comes in at tens of grams, it's probably going to be difficult to find the causal switch because of this self-catalyzing property, right? So there's a seed formed in terms of abnormal clotting, and no one is going to argue the case that when you get exposed to COVID or you get vax injured, there's a demonstrable phenomenon of amyloidogenic microclotting. Okay, so people will, unfortunately, there isn't an automated way to do this type of investigation. It literally requires someone manually looking through a sample and dyeing the sample for amyloids with the accepted validated dyes that are used for this type of study, and basically just counting how many objects that they see in someone's blood sample. And if so, you know, just to give this, I was to call it scuttlebutt, but like the discussions that sort of go on with people who I know are working in this space, is that now it's more and more difficult to find people who just have normal blood. Literally everyone, but the problem is that when people, the samples they're looking at is people going and trying to get treatment because they don't feel well.
Health issues,
and so it's difficult to say.
We don't know what the population-wide distribution is of blood. We are assuming it. And this is what you've done. You basically got the samples from the cadavers, and you've sliced them up and put them into a machine. And I think probably the most concerning part was that in 7 hours you were able to get to show that it was amyloidogenic, that it could be potentially seeding, like let's say someone was exposed to that somehow, that they could develop some sort of preogenic disease. Is that what I'm
so let's work through the data. So I will try and do a PowerPoint presentation, right? And we'll work from there. I just want to make sure I'm pulling up the right one. Yeah, I can't speak to how much of this was deliberate versus how much was something went wrong. They did have an accident, and then we end up where we are today, which is a very, very large disease burden, and the authorities being less than helpful in dealing with the situation, because one, the interface with the public is going to be, this is unfortunate, but a lot of doctors are in debt because of medical school. They're often very ideological in their thinking, in terms of the power dynamics between them and the patient, and there's all sorts of obstacles in the way to getting the clearest answer, which I think the public is owed at this point, right? And after they've lied about the origin, after they've essentially engaged in mass psychological operations where they quote unquote lockstep people into a public health measure based around gene transfection, and suddenly we've got a emergence of disease forms that, because of the nature of the disease causing agent, it's just going to manifest in people differently, and you're going to have autoimmune disorders, cancers, cardiovascular, etc. All of those can have a proteinopathy, meaning a misfolded protein as the causal part of the disease that's manifesting. I hope that didn't sound too convoluted.
And then people will not know the point of origin of it.
Right,
which is what we're working on now, that if we find the point of origin and the mechanism for how these things are being manufactured in the body, we potentially are looking towards some relief.
So hopefully. Now, look, I can say that with some confidence. Now, I have to maintain scientific neutrality in the current environment, okay? But I'm fairly confident in the results that I am seeing in the groups that I'm collaborating with, that there are techniques available to lower the burden of spike protein, that there seems to be a reservoir of spike protein, and the body is reacting to that, and it's not just immune dysregulation at this point. The peptide itself, as we'll see, is causing this buildup of toxic peptides in the body itself, and though that buildup of toxic peptides potentially leads us to our good friend here, this amyloidogenic clot. Okay, so I'm going to share screen, and the same techniques that I used to examine the vials then, Raman spectroscopy, scanning electron microscopy, form part of the backbone for looking at these peptides here. Okay, and I wish to distance myself as much from the orthodox institutes that gaslit the globe as a whole versus the grifting side of the social media networks that are looking for well, they're just making up lurid BS in order to attract an audience and basically maintain a funding. So what are we looking at here? So on the left hand side, that's the arrival of the first samples that came to the lab. Same patient. One on the left is just preserved in a little bit of alcohol and argon. The other is coated in formalin, and we're going to start by just looking at the gross anatomy. What happens when I crack open the vial, pull it out, and what does its physical properties feel like to me? Okay, and so I've done this live on stream, right? People can go back and look at the streams that I have done on my channel, and you can watch in real time as I do this and do the analysis. Now, I can't do everything all the time under camera, but I try to do as much as possible. And just as a heads up for people, just because of the amount of time that I'm having to put into the lab, it's too much for me to set up a full stream like going out to Rumble, etc., for people to watch what I'm doing in the laboratory. But what I do is using my Discord community, I will go live in there, and then everyone who's joined in there are more than welcome to watch me working in the lab. Now, people seem to like it for some reason, and I'm happy to show what I'm doing, which is just, it's opening open doors, taking stuff off with pipettes, and just running stuff. There's some work on animals that I've done, but I can't make a full stream out of that. So if you want to see those details, ask to join the Discord, okay? And that's first point. So yeah, you're looking at a standard dissecting microscope there, and basically it's a pair of forceps and essentially hypodermic needles that I will use basically to pin the sample under the objective lens so that we can look at the object at hand. This is not through the microscope. This is me just taking a picture with my cell phone, but now we'll start to look. Well, let's talk about what it is that I see when taking that sample out. And yes, it feels rubbery, and the closest that I would describe it in terms of how it feels is, if people have ever eaten like crab sticks, fake crab sticks, it feels like that, right? But maybe a little tougher because a crab stick sort of peels easier. But in terms of tensile strength, it's close to that. Now, although there's resistance, I could pull that clot apart if I wanted to with my fingers. Okay, but it does have a degree of elasticity to it, and it does appear unusual just in its presentation. Okay, just to my eyes and just my experience of working in the life sciences. Okay, so this should say 1 millimeter. I'm sorry. And this is now looking through the scope for dissection, and you're just getting a close-up view of the surface of the clot. And what would I say looking at that myself? There seems to be a striated appearance to it, right? So I don't know how clear that is to people.
Yeah, it looks pretty clear.
Right. And there are also sort of major creases and folds.
So how would you have expected something like that to look instead?
Look, the only clots that I've ever had to deal with before are just the immediate ones after cardiac perfusion. It's usually the jelly-like clots that literally would just, if you handled them, just sort of break apart very, very quickly. Like I say, this is proteinaceous, and it's almost the rubberiness of it is almost like the brain after I've done a transcardiac perfusion with saline and ran some formalin through it. That's kind of how it feels, right? But obviously the brain being more lipid in nature, it has a more spongy feel. But in terms of sort of tensile strength and just sort of feeling it, fingers and what have you, think crab meat, or if you've ever worked with perfused tissue and formalin brain tissue, it's close to that. And obviously the color is important, because the color will tell you some idea of its constituents, and the normal or the majority of clots that come out are ones that essentially are aggregating red blood cells and platelets, and a loose, well, if they're immediate rather than sort of impacted ones over time, the fibrin is only just having chance to form, and so it's not forming a more solid mass than that I would usually see. So I see, in my experience, I've seen immediately forming clots. I don't have experience in the cath lab pulling out a clot that's been building up over decades. Okay.
So
Something.
while we're talking about the clot,
Hmm.
are you in agreement with the analysis that it has high levels of, what was it? Phosphorus?
Phosphorus, sulfur. So I'm lining up these. I know other people have done the ICP mass spec. Okay. I'm not going to say that they were wrong in their analyses, but what I want to do is I'm going to do this as independently as possible. I will run all those tests and more to try to determine exactly what it is that we're dealing with.
Okay.
And what I will say is that where I've done mass spec type measurements, I have not found anything that would point to there being metals, tin, heavy metals being part of the picture, right? Everything just looks, well, not just, everything looks like protein. And in this instance, in order to work out whether you're dealing with a misfolded protein, there's a whole number of steps that you have to go through in order to say that you've ticked all the boxes to say that you're confident that it has the properties that we ascribe to misfolded peptides that we have been looking at for decades, okay, and associate with different diseases. Now, so to your question is there a overabundance of phosphorus? And phosphorus plays an important part in the body. So there's a metabolic pathways called kinase signaling, and control of phosphorus along those pathways is critical for maintaining bodily health. And to give a sort of example that relates to COVID, one of the earlier, more spectacular observations was that cells infected with COVID began to take on very different cellular morphology than you would normally associate with. Let's just say we're talking about lung tissue. Okay.
Yep.
So cells within the lungs get infected by the virus. Viral infected cells undergo morphological changes in which they develop pseudopodia, which basically means that they develop branches and tentacles. And that switch in morphology is a process or dysregulation of kinase pathways, and that involves phosphorus signaling through multiple steps. They're incredibly complex pathways that essentially, in this instance, are interacting with actin, and which forms the cellular cytoplasmic structure, if you like. Right. So a cell has actin in it, and actin helps maintain the shape of the cell. What's happening is the kinase pathways are changing. Actin is forced to change, and it then starts making these pseudopodia, and the pseudopodia act as an attack vector for viral vesicles to travel to neighboring cells. Right, there's fantastic work out there, and it's going back a few years, but where they literally can show viral particles traveling along and through these pseudopodia tentacles and piercing and infecting neighboring cells. Okay, so does that relate to what we're seeing here? I'm unsure. Again, causality and interrelations is a complex topic, man. You can spend decades trying to work at something like a problem like that. Okay, so I don't have answers for this particular issues, but let's just give them the credence and say that the ICP mass spec that has been done has found a large phosphorus signal. Beyond that, I'm not sure what I can say or what that means really with respect to the final form here, which is the amyloidogenic stacking and misfolding of protein in this instance. Okay, because I haven't done the analysis yet myself, and I want to make sure I've run all through that, through all samples, and then I'm going to have a better idea of what we're dealing with. Okay, yeah. And that's not to dismiss the work that others have done. It's just
you might as well confirm it.
Yeah, I want to be clear in my mind, and I want to have done the experiments just so I know exactly what's coming out, and I'll have it done in the coming weeks to months. Okay, but we already have enough to sort of move on from there. So we're looking, the sort of gross morphology here. The obviously the color is different to normal clots. It has this striated appearance, and I think a lot of the sort of larger morphological features, I think, just come from the imprint from the inner lumen of the vessel in which the clot is forming. Okay, so now I'm basically just taking, if you go back and watch the videos, I don't know, what's this is up here, I need to change this, and I'm taking a scalpel blade and I'm just slicing through that segment, and what you can see is that it's not a tubular structure. So people have claimed that what the morticians are pulling out are blood vessels, etc., and this shows that's not the case. These are it's a solid deposition of peptide that well, even saying peptide prior to me looking at it was people would have challenged that, because the test hadn't really been done, and the people doing the testing, but they would say probably weren't qualified to be doing. So it's just another look. Let me just go through this and change this. And so that's the gross morphology of just one sample that I've pulled out. So it already looks different from what most people would imagine a blood clot to be, which are usually sort of red in nature, and again, depending on the age of them, their density and stiffness will vary. Again, it's very, the color is off here. I think it's just a consequence of my camera and the angle.
How wide are they? Like how,
No, so you've got the scale bar there, and
okay.
sort of 2, 3, 4 millimeters wide, 2 millimeters thick. I think it depends the vessel in which they get. Again, I'm getting them in, sort of 10 mil tubes. I've seen much bigger shown to me on camera, but this is enough for me to work with in terms of doing this type of anatomical analysis, molecular analysis, and testing against or trying to use it to develop a test platform in animals. Okay,
well, certainly they have no place in the blood, right?
Oh yeah, for sure. Someone who, like, the concern at the moment is there a silent wave of people carrying around this this blockage, or this clot-like phenomenon? And again, I would go back to what I was saying earlier, is that what I think has happened is the system has absorbed it. It's basically recognized and stated that, yeah, we know that there's a clotting phenomenon. The primary diagnostic criteria for COVID is systemic coagulopathy, meaning you've got clotting occurring across the body. Okay, now that in a lot of cases looks like the microclotting, the amyloid microclots, and usually 100 micrometers in length. Okay, sometimes bigger. sometimes smaller, but on average about 100 micrometers. These larger clots, again, I think have been absorbed by the system, and if the powers that be worked so hard to obfuscate to the public the source of what COVID was and the nature of the public health measure that was enforced upon them, you don't think they're going to be just putting the brakes on in the surgical centers and cath labs that receive these patients and basically just saying, look, man, just make a note of it and then just throw it in the trash. And if someone is unfortunate enough to be severely injured or dies from that, that basically gets labeled as a cardiovascular event and potentially COVID linked, right? For sure, they're not going to be saying anything about vaccines in this instance. Okay. Now, I'll state for the record here, I have to remain agnostic as to the cause of this phenomenon, because it's clear within the literature that spike protein in and of itself can form or can initiate and catalyze a misfolding of fibrin by itself. Now, but I would say, does it help if suddenly you expose people and dump in billions of molecules of reactive epitope that we now are working towards it causing a misfolding of fibrin, a pathological misfolding of fibrin and the clotting process, right? So why are these forming? Because of the misfolded nature of the peptide. Enzymes that would be involved in their breakdown, like thrombin, are unable to latch onto them and begin the degradative process. So in effect, they'll hang around, and because of the type of disease mechanism that we're dealing with, which is the amyloid templating of normal healthy peptide into the abnormal form such that it sticks together and then begins to aggregate into bigger and bigger structures. Now, what's the basis for that? It's the current hypothetical framework is, and I think there's a lot of evidence for this. It's not new in the fields that I work in, is that there's a development of what's called beta sheets that become hydrophobic, and those hydrophobic beta sheets will then stick together like Lego bricks and just form larger and larger fibrils. Okay, so this was basically the extent to which people had looked at these clots, right? That, like I said, there'd been some mass spec analysis done, but the rigor required to look at them beyond this hadn't been applied. Okay, so in my lab we have the capability of doing all the histology and more to look at the microstructure of the microscopic structure of these clots. Okay, so how do we do that? This on the left is called a freezing microtome, and what you do is, well, there's two types of microtome primarily. One is paraffin fixed and embedded, where that's a good way of quickly scanning through a section, but it's not so good for antibody work. I went for a freezing microtome because I thought I was going to be doing far more antibody type anatomy with respect to the work I was doing with recombinant peptides and any modeled type pathology that we could induce in animals. Okay, and you know, the best laid plans of mice and men as such, I've had to pivot around the original aims and directions that we're going for, and use the setup that we do have to go to try to analyze these this phenomenon. But what this machine does is basically it'll hold so you see the orange block in the middle on the right here, right? So this will go down to minus 30 degrees Celsius. It's held in a special type of gel, and there's a very, very sharp knife, and it will step forward a set number of micrometers every time I want to cut through the tissue, such that you get even slices of that tissue so that you can put it under a microscope. Okay. And that's what you're looking at here. So I've basically taken 3 slices and put it onto a standard microscope slide. Now, the thing that what should stand out to people is what look like here, what looks like whole tissue, right? When you start cutting it at the level required for microscopic investigation, you see cavities and small holes that are visible to the naked eye, right? So that's essentially now what we're going to see as we start the process of going down in scale from, you know, the light microscopy into the realm of electron microscopy. Okay, so this is 4 times. This is generally the lowest objective magnification that you get in a on any microscope, whether it's a $30 kids microscope off Amazon to, you know, the microscopes that cost thousands and thousands of dollars and can do all the fancy tricks with UV lighting. And so what do we see? Yeah, there's it's not a solid planar piece of tissue like you would see if I was cutting, say, brain or muscle tissue or any other organ tissue in the body. It would be very, very unusual to see this type of porousness, I would say. Yeah. I guess that works. And those with a keen eye will start to notice that there's a fibrillar nature to the material, right? And this has been cut at 20 micrometers. Okay, that's how thick the section is. And so this is now at 40 times magnification. That's actually the maximum that I have on this scope for UV investigation. But it's quite clear when you're looking here that, yes, we do see fibrillar type structure, and this would concord with it being fibrin. Fibrin forms fiber-like networks that are basically designed to hold platelets and red blood cells into clots. That's its job. And you have to think of, in biology, sort of form equals function, right? So its job is to form a network that acts as a sort of trap for normal clots, and the you need that so that you don't bleed out should you, I don't know, step on a nail or, you know, whatever. You know, there are, if you're a hemophiliac, etc., a paper cut can be deadly, but you know, you need a clotting response, and fibrin forms part of that. It's one of the major components of your clotting that's occurred here. Now, what's of interest here is that there is very little or no to identifiable red blood cells or platelets or any of the other normal tissue types that we would associate with a clot in this instance, right? It's just this very fibrillar form, and the color that you're seeing at this level, which is just normal white light being shone through the specimen, matches what you're seeing at the macro level, which we looked at a couple of slides previously. Okay, now, okay, so this.
So is this stained or not?
No.
So this is what when you actually cut it and you put it under the microscope, it has that green appearance.
It's not green per se. Like, to my eyes, it's more a it's a cream color. The green that you're seeing is refraction of light through the fibrils, because essentially they're translucent. But if you look at more dense areas, it's essentially more akin to the color that you're seeing in the larger samples. Okay, so so with light microscopy, you get some idea now as to what the structure is that we're dealing with. So we've gone from gross morphology down to the now you could go to 100 times with light microscopy, and you'll get a blurry view of some of those fibrils. Okay, but what we want to know is what's the nature of the peptide chemistry that makes it up. Now, a way to do that is through all manner of different stains, but because I've been banging on about amyloid peptides, prion peptides, etc., for years now, I've been asking, and it's how I managed to get a hold of these specimens was just by people who were working on them. I said, you need to be doing amyloid staining on those tissues, because you can't say anything about their functionality until you do that. Okay, and that's how I sort of managed to get in contact with Richard and get him to send samples. Okay, so what do we see when we look under the specimens with? So you can switch to ultraviolet light, and that's how we use, well, the primary dyes that we use. So the legacy dye was something called Congo red, where you would have to look at it under sort of polarized light. And if you see, and the problem is that that effect is is prone to a degree of subjectivity. And because you're looking for something called bifringence, which basically says, under polarized light, do you see a halo around the structure that you're looking at? And if you do, that would indicate that you're looking at an amyloid-like structure.
Okay.
Using more, well, I say more modern, but they've been around for a long time. But the other approach is to use analysis under UV light and look for absorbent spectra and the response to specific dyes that have an affinity to amyloid structures. And so what does that mean? So I talked about the beta-pleated core that we think makes up this the primary structure of the amyloidogenic peptide. And what these dyes do is, between each layer of the beta-pleated sheet, they'll sort of insert themselves, and they'll go in there and they'll fill as many spaces as possible between each layer. And then under the UV light stimulation, they basically change conformation and will basically give off a wavelength that's hang on, so if you hit it with 480 nanometers, I'll say they will reflect light back to you at 450. I think something like that. I'd have to go back and just refresh myself on the details there. But and so one of the first things that I noticed was if I just looked at that tissue under UV light, so that's what you're looking at on the right hand side of the screen, it auto-fluoresced even without putting any dye on. Now, there are some molecules in the body when you're looking at tissue that you'll see when using UV microscopy that will light up, and we know that they exist. Okay, and I don't know, fusin is a very common one that you'll come across, and this, but I've never seen whole tissue sections without staining light up to UV light like you're looking at there on the right hand side of the screen, right? It's basically saying THT thioflavin negative, right? This is at the lowest magnification and doesn't, without doing anything to it except cut it into those 20 micrometer sections, putting it under a microscope, and then hitting it with UV light with a blue filter that will then excite in the green range. Okay. When you drop thioflavin onto the slide, onto the specimen, suddenly what you'll see is that specimen will avidly take up the stain, that you start to see far more features begin to appear in the image that you see off the microscope. And what you can see is basically at this level of magnification, you're looking at small sort of punctate dots all around the sheet of material that's been taken from the microtome onto the slide. Now, we can sort of get into discussion about exactly what that means, but what I would, my first, and the way that I look at it is that when the amyloid is forming, right, it's going to start stacking, and where it stacks at areas of high concentration, you're going to get higher concentrations of dye, and that's what I believe you're looking at here, is all those little dots are areas where you've got more stacking relative to the more lighter areas of the image. Okay. I know that's a lot to sort of throw at you. Do you have
And the implications of all this?
That you're dealing with any systemic amyloid type disorder is considered along the sort of scale of disorders that you can turn up to a hospital with as being one of the more serious conditions that you can develop, right? And people who have light chain amyloidosis, right, do not live as long. There's very little in the way of treatments, and as they progress through their illness, the reason that they die earlier is that the light chain amyloids penetrate into all their organs such that the organs shut down because they become gummed up with this amyloidogenic peptide. Okay. Now, if
so it kind of explains a number of things that people might be seeing out there in the world.
Yeah, so this is something that I would encourage people to maintain, which is that because of the nature of this type of disorder, it can manifest in a number of different ways. Okay, cancer is one, neurodegenerative is another. In this instance, if your circulatory system is filling up with this undigestible amyloidogenic peptide, you're going to develop all manner of cardiovascular and thrombotic type tissues or diseases. And again, if there's no desire on behalf of the authorities to look at this, because essentially the blowback would go to them anyway. The buck stops with them in terms of how the public thinks, right? And if people have lost loved ones, either due to exposure to this synthetic virus or due to just to force public health measures using novel techniques that have never been deployed en masse before, where you've made the body express this toxic peptide that can act as a seed, right, to catalyze something into the, well, should be wearing gloves, where you're getting grams of material post-exposure.
It's pretty dark. It's very dark.
And all of it fits under the umbrella of proteinopathy. Proteinopathy means misfolded peptides, and there are hundreds of identified peptides now that are implicated in any number of diseases. Now, I would just encourage people to think about what would they be looking for in terms of a battlefield incapacitation agent. You can't wait for the opposing side to develop tumors. You want something that initiates a quick knockdown of their fighting effectiveness. And in this instance, if you can gum their blood up with a non-digestible clotting agent, and clotting agents in and of themselves are a biowarfare, they're a chemical clotting agents, right, that they can deploy.
I mean, this has some sort of implications for, well, I guess we haven't tested yet, but whether these seeds, if we want to call them, are present in saliva, blood.
But that's something
feces.
we'll get to. Okay.
Yeah.
So we haven't finished yet. We've just started the analysis into what we're looking at. Okay. So okay, we can start looking at the limits of the light microscope using UV. And hang on, let me just check that's not the wife. All right. Let me just do this. Right. So you're just looking at higher magnification here, negative versus positive stained tissue, and you're just getting a closer look at the sort of punctate lesions, which I would hazard a guess, or not hazard, it's not guesswork, hypothesize are more aggregations of beta stacking. Okay. And can we answer that question? Is there a way to determine if there's beta stacking involved in this structure? Okay, so what we can do is you can then increase the magnification that you use. So you go beyond light microscopy. This is the scanning electron microscope we have. And now we're going to go down in where you're going in tens of magnification. Previously, we can go in thousands of magnification, power of magnification. Okay, and so what are you looking at here? So using the microtome that you saw previously, I'm cutting at its limit basically, which is approximately 5 micrometers. There's going to be some jitter in that just because it's at the limit, but I've taken the thinnest cut possible that I can of the tissue, and then tried, and it's very hard to get that off and laid out in as even a form as possible, just because it's so thin. Okay. Mm-hmm. But and so it has a somewhat sort of folded appearance on the bottom half of the image that you're looking here. Above that is just the glass slide itself. Okay, and this is at 25 times, so this is in the range of a normal light microscope. Now we're going down to 250 times, and you're going past what a light microscope can reliably do. And what do we see? Well, we see what looks like, again, sort of fibrillar type structures that we saw at the light microscope level. And so like the way to think of it is essentially sort of fractal, right? As you go down through the scales, okay, or power of magnification, right? And so you'll see a sort of gross morphology, and then as you sort of see coherent structure, when you get to where you see coherent structure, which was the fiber matting, and then what looked like punctate aggregations, as we increase the power, as above, so below, you see the same sort of structure still there. At this level, basically at this power of magnification, basically you're zooming in to like one fiber length or cross section, right? So light microscope is showing you a mat of fibers. Electron microscopy is now showing you what one fiber looks like in that mat of fibers. Mm. So that's 250 times. This is at 1,000 times. So again, what do we see? It looks like a crisscross network of peptide, but there's also sort of bulbous aggregations. Okay, and this will become important as we go through the next slide steps. Okay, and yeah, again, just to reinforce to people that what you're looking at here is at what was at light microscope level, one fiber, right? So I just want to reinforce this point, right? So if I zoom in, you're basically looking at one fiber. I don't know if my cursor is on the screen or right. So you're basically looking at one fiber here, right? Go down here, zoom.
It's amazing what the machines can show you.
Yeah, and
there doesn't look like any nanotechnology there, Kevin.
No, this is and it's been incredibly frustrating because I've had a sort of public role in not just pushing it back against the orthodox and the corporate quangos, etc., but fighting against the stupidity that sort of emerges on the internet. I don't want to say stupidity because that's not fair to some people who have concerns around things like synthetic biology. Synthetic biology is a train that's barreling down the tracks towards us just as a consequence of the development of technology across multiple domains. Okay, I don't think that you're seeing that in this instance. You're seeing something else. What you're seeing, again, just to reiterate to people, is the deployment of amyloids across they've turned the planet, the whole planet became a battlefield in this instance. And whether you could perhaps argue that this was a test for them, right? Do these make good weapons? Right? When you deploy them, can you knock out 30% of a fighting force if they don't know it's been deployed, and they all develop blood clotting, sort of instant blood clotting, such that their oxygen carrying capability is reduced by 50%, right? So like I say, you see this crisscross mesh of proteinaceous fibers. Now, you look at most structures under a electron microscope, and you'll see something similar to this, and that you could say is the normal features that you would expect to see. But in this instance, when we go down now, right? So this is at 1,000 times. Okay, but when you go down to 5,000 times, what you see here, and it's only because I'm familiar with looking at these structures that this was standout abnormal to me, right? There are very clear nodular forms on these peptides, okay? I don't know if you can see that, right? So where my cursor is here, right? They sort of stick out. And if we look at this primary branch here, what you see is fibrin should be a long, smooth, rope-like peptide that essentially just overlays itself to form the network around which platelets and the other tissues that form a clot aggregate. Okay. Here, what you're looking at is so you're looking at very abnormal structural properties that are standout to someone who's familiar with looking at peptides in and of themselves. Okay. And those are these nodular forms. And also what I would point out to people is that pay attention to this thicker filament, right? And in biology, often what you see, and the best example that I can give for people to think about is often when you look at a tree, a tree, as you look at the trunk, has a sort of twisting effect to it, right? It sort of
Mm.
starts at the roots and it sort of has a rotation to it as it goes up to form the branch area of the tree. But the trunk often, if you pay attention to it, you'll see has a rotation to it. And generally in biology, I want to say it's a right-hand rotation. It's the right-hand rule of thumb, and you can get into all sort of metaphysics around electrodynamics and how that relates to the body. But in this instance, what you're seeing is you're seeing a faster twist, so rotations per unit of distance, than you would expect to see in normal healthy tissue. So imagine taking a piece of, if I have a piece of string available, but I don't. But if you took like a normal piece of rope and then you started to twist both ends, what happens to that rope? It starts to tighten, right? And then it'll sort of it starts to deform, right? Just think this simple thought experiment. Take a shoelace, start twisting it at either end, and eventually it sort of shortens, and the normal turns that you would get compressed per unit of distance that you would use to measure, right? And that is one of the primary features that we use to describe in the scientific domain whether a peptide is normally folded or abnormally folded. Okay, does that make sense? And again, I'm throwing a lot at you because.
So basically, you're saying here, yes, what you're seeing is it's proteinaceous, that it is. Is that what you're saying? That
It's
you're
just protein, right? It's just protein.
Misfolded protein. Is that what you're saying?
Nothing else in there. It's just a big slab of misfolded peptide that is inherently obvious if you have the tools to probe it. The tools, well, the analysis that had been done prior to this, whether it's people looking at it and seeing there was no analysis done. There was some mass spec done, and unfortunately the initial mass spec was done by Mike Adams, and his just reputation is toilet paper on the internet with respect to doing anything scientifically consistent. Okay, and again, what he does is he's trying to drum up audience numbers, and so we'll immediately go to the more outrageous descriptions. And he was talking about it having circuit-like properties. No, you're looking at just misfolded peptide, just lots of it.
Yeah.
And the reason there's lots of it is because of the peptide that's involved, which is fibrinogen to fibrin, which your circulatory system is full of in preparation should you slice off a finger using your chainsaw of a weekend, right? Right. It's just the nature of the
Your body's got so many ingredients to make this if it misfolds. I mean, I think that was always a concern of mine. This whole nanotechnology idea was, where is it coming into the body to make such a huge amount of whatever, these.
Well, the reason it's such a huge amount is because you have so much fibrinogen per mil of blood. Okay, it's like in blood you'll have red blood cells, platelets, and one of the primary, you have albumin, and then one of the other most common peptides are those there for clotting, fibrinogen, which is there converted into fibrin. Okay, and that forms the clot. And the reason you have a lot of it is sometimes, like I say, you can have a massive wound and the body needs to pile loads of this peptide in to plug the gap, otherwise you bleed out, right? And so there's nothing unusual here. That's the wrong word. There's nothing spooky or mysterious here. It's the fact that there's a misfolding of fibrin to fibrinogen, fibrinogen to fibrin, the clotting peptide that is causing this mass to appear in individuals. That's what I would posit and hypothesize. I haven't been able to witness firsthand the extraction of these clots, but I'm trying to make sure that whole chain of investigation and necessary legal steps is taken care of, so that we can we can push forward. All we can do is just, you know, one of the few wise things that my PhD supervisor said to me is just always put your best foot forward in this instance, especially when you're dealing with what is essentially an unknown that exists at the edge of our common understanding, which is the whole point of doing scientific investigation, right? So there we have, in this instance, black and white, prima facie evidence, visual evidence that you can see that it's misfolded. Okay, now, well, show us that it's misfolded. Where's the why am I missing slides there? Oh, I know, wait, I'm looking at the wrong presentation. Yes, let's go here. So
is this what we would see under the scope in terms of other neurological diseases? Or is it something?
well, you just don't have this volume of material to look at.
Oh, okay.
Right. So even in something like Lewy body dementia, Parkinson's, etc., when you look at the tissue, when you've stained, you use the same dyes that I've used here, you'll have little punctate spots that are essentially intracellular inclusions, or often, or depending on the disease, will sometimes be extracellular, but encased in the myelin or in the extracellular space. You know, the characteristics between different proteinopathies are varied, nuanced, and essentially you have to spend a lifetime studying them in order to have a good image in your head of what they are. Now, I'm confident with respect to Parkinsonian-like neurodegeneration, because I've spent 25 years investigating it and working with it, and I have a extensive work, just hands-on experience with what that tissue looks like. I can cross-reference that to other disease states, but do I have the type of in-depth knowledge that I have with respect to Parkinsonian-like neurodegeneration? No, I don't. I can understand what someone who has spent 25 years working in Alzheimer's is saying, because, you know, we've come to a common understanding that there is this amyloidogenic stacking of hydrophobic beta-pleated sheets that occur in proteinopathy. Okay, now, in the summer of 2024, this manuscript came out. Fibrin drives thromboinflammation and neuropathology in COVID-19. Now, this was a very, very good paper. I encourage everyone, please take a snapshot of of what's on the screen here. It's an open access manuscript. And
I don't think it's on the screen at the moment.
It's not sharing?
Not that one. You've still got the calamari
wait.
plots. Yeah.
Yeah, wait a second. Zoom.
I'll put a link to that in the article as well.
It's not giving me where's the little window that, oh, it's up here. Sorry, it's on a completely different screen. I'm going to stop the share, right? And then I'll put this one and Zoom, share, this one. All right. So, you know, we were looking at this slide, okay? And so I was saying that there was a manuscript published. This was from University, California, San Francisco, 2024, and title being Fibrin Drives Thromboinflammation and Neuropathology in COVID-19. Now, and this is going to present a degree of, how do I say, well, yeah, I'll be brutal. There's going to be a degree of cognitive dissonance here, as people who sit and have their own view as to what's caused what, are going to get challenged by the just extant data. And like I said earlier, my job is to remain as objective as possible in the scientific investigation, such that I'm not led astray by my own confirmation biases. Okay. And what this manuscript has shown is that, and very nicely, right? So that's behind that is the image that we were looking at before. If I sort of zoom in, right? So here you can see an example of what on the left-hand side, human plasma, fibrin, and you can see, like I said, it has this smooth rope-like appearance. If you expose it to the spike protein, what happens? Oh, it starts developing abnormal twists and little bumps and nodules that we were looking at on the image that's sort of in the frame behind, right? And we were looking at on the previous slide. And so they're looking at a proteinopathic form of fibrin in controlled experiments. And look, I did my PhD primarily at UCSF. Okay, very, very high standards of work come out of there. A lot of other deranged stuff does as well, but you know, in terms of the validity of this research, I'm fairly confident that they would have gone through all the steps required to ensure that it's up to the sort of highest scientific standards available. And I know people have just a distrust of that, and you're sort of you need honest brokers and scientists to be able to tell you, okay, in this instance, this looks legit, and this says nothing about the source of COVID, etc. It's just looking at the pathology that we can see when there's spike protein exposure in this instance. So for all the people who think that long COVID is just that's very obviously not the case, because there was a whole cohort of individuals that got impacted by the long-term sequelae of COVID infection prior to them even deploying vaccines. I know many people who have been just impacted severely post-infection, and they made a point of avoiding vaccines. That's not to say that I don't know. I know many people as well who have been injured, but basically people have sort of got it into their heads it can only be one or the other, whereas you know the world is a more complex place than that and more nuanced, and so you're required to sort of, you know, you've got to take the evidence that's out there, and you can't just eject something because it doesn't fit your current mode of thinking. Okay, now what we can say is spike protein in this experiment is giving us this misfolded nodular-like form of prion protein. Okay, now it's not a leap of the imagination to say that, well, if you go and gene transfect people and we have long-term expression of spike protein because of the nature of the way that they made those products, right? Methyl pseudouridine to prevent breakdown, and they've already come out and admitted that because of methyl pseudouridine, you get a frame shift that can occur, and basically you get a noisy expression suite of peptides that get released. And so why do you see that nodular bumpy surface? I would argue vociferously that there is a epitope signature that's part of the spike protein which has amyloidogenic catalytic effects aimed at the prion protein. Now, Dr. Stephen Quay in I should see if I can find that, but he put out a preprint looking at the N-terminal domain of the spike protein, this is an important point actually that we should bring up. Internal domain. Give us a node. Yeah, it's this one. Now it's sharing application, right? Rather than, if I drag this over, does that, you see that?
Not yet. No, just keep going.
Yeah, it's I have to go through the whole thing of sharing screen because I keep sharing application. Let's do this. Stop share. Let's share. Leave it's that one. Yes. Share. All right. So, Dr. Stephen Quay is I know people who know him well, but I've never had the chance to really speak to him personally. But what people have said about him and the work that I've seen him produce, I don't have concerns about his scientific motivations. Okay. So this was a manuscript that he submitted, put up onto a preprint server, but I think is of critical importance. So the thromboinflammation and neuropathology sequent motifs of SARS-CoV-2 spike protein appear to have been engineered into the virus. And so if something's been engineered into the virus, it's also been copied into the vaccine itself, because basically they've gone for a codon optimized copy of what the spike protein is, turned it into a synthetic mRNA molecule, and then put it into the population at large, okay? And basically bypassed immune barriers. So a landmark paper, and this is the paper that I just showed you, Fibrin drives thromboinflammation and neuropathology in COVID-19, was published in August 2024, that concluded the mechanism of the thrombotic and neurological symptoms following a SARS-CoV-2 infection, often called long COVID, is attributed to the binding of fibrin to discrete proteins of the spike protein, specifically 3 N-terminal domains. This paper is a high-impact publication with greater than 110,000 views, placing it in the 99th percentile. Here I examine the regions of the spike protein that bind to fibrin, fibrinogen, or both. The N-terminus of the spike protein contains the 3 strongest binding peptides, and surprisingly, these regions are also the 3 insertions in the protein sequence that are unique to SARS-CoV-2 and not found in natural sarbecoviruses. So if people have the stomach for it and the patience for it, if you go back to the beginning of you shouldn't call it a pandemic, what we were talking about is a mass casualty event because of biowarfare programs.
Yeah, word's important. Yeah.
Yeah, because if you call it a pandemic, it disarms the cognitive apparatus as to think of it as something natural. Okay. A conclusion that the pathophysiology of long COVID is based on the insertion of spike protein motifs with sequence homology that mimic HIV GP120 protein motif properties, and that these SARS-CoV-2 motifs are not found in sarbecovirus subgenus, strongly suggests that these inserts were design features in the synthetic assembly of SARS-CoV-2, meaning it was a deliberate insert into the biowarfare agent to initiate a specific effect, the targeting of fibrin, such that it begins to misfold and you develop the sequelae of long COVID, which is characterized by extreme fatigue, neuropathy, and what's essentially a chronic a chronic lack of oxygen perfusion of deep tissue. Okay. Let's see if I can get back to PowerPoint. So so from these calamari clots, okay, you can just reject all the nonsense that has been said about them And using the correct tools, you can look at it and see that it's a misfolded peptide that lines up with and has a causal mechanism related to what's called the N-terminal domain of the spike protein in and of itself, right? So contact between one and the other will cause this misfolding, and because of the nature of fibrin and just the amount of it in the body, it's going to cascade, and the spike protein could be long gone.
Yeah. So, I mean, I know we can't possibly answer it based on what we know about this. It's still kind of, I would say, experimental. Yeah, we're sort of going into the great unknown here. But what is the prognosis, do you think? Like, what sort of timelines are we looking before we're seeing a lot more injuries from this?
Well, as a endemic pathogen, it still does the rounds. My family just got a big dose of it after Christmas, and you know, we had clinically identified influenza prior to that, hit all the kids, etc. And a few months later, COVID, and they were distinct illnesses in and of themselves. So for those that want to say it's just flu, they can. I'll keep my language.
Well, we can hope we can move beyond that. I think there's enough evidence to say that all of
Well,
that.
it's overwhelming if you want to look at the evidence, right? I'm not even finished yet. There's more, right? So you asked about the elemental analysis, right? And so this is a technique. So when you're using scanning electron microscopy, you can do something called EDX mapping, right? Which is a electron dispersive X-ray mapping. So you're hitting it with a electron beam, and you can get a sort of form of mass spec, and it'll tell you what that structure is made of. And what do we see? Calcium, oxygen. Silicon is the glass slide on which it's in. Chloride and sodium is the buffer in which the peptide was kept and processed. Magnesium, aluminium. Aluminium possibly because the stage on which the slide is sitting is made of aluminum. Calcium, phosphorus, sulfur, and nitrogen. Now, do those ratios seem abnormal? I don't know. I will do the ICP mass spec myself, and we'll get a measure, and I'll get a measure across many, many more individuals than has currently been done, and we'll see. And I'll see if I can sneak some regular clots in there and see
I
how
mean,
they
if they
can.
were abnormal, you would think, let's say, theoretically, the phosphorus was high. Wouldn't the body have to, find that, like, wouldn't it be causing, where does that grab it from in the body in terms of balance and
Well,
homeostasis?
it's scavenge it from anywhere, right? That's
So
yeah.
it's not like you would say the food intake is going to make any difference whatsoever. People.
Well, this is a good question, because we don't know if there's some environmental factor that may be another trigger in the formation of the macro formation of this phenomenon. I can't exclude that at the moment. And
People were eating high phosphorus foods, like, I know we have.
I don't know if it would be that in and of itself, but the I'm just gonna just say I don't know at this point, right?
So
And
we're looking at, we could have multiple triggers because we don't know why, say, some people who are exposed to it within a certain family react differently.
and that comes down just to the individual's own genomic makeup, environmental makeup, their health status, etc., etc. There's innumerable factors, and one of the issues that I find all the time is that people will have a binary way of thinking about any problem, which is if that happened, then so if they got COVID, then they should get XYZ, when we know that no, that's not the case. Some people will develop all the problems, and some people just get it and it's not an issue, right? And
Or could be a delayed reaction, perhaps.
or delayed reactions, and there's the clinical picture is so large, and again it's going to come back down to the honesty and veracity that we can put into the institutions and the clinical front line as to what they're seeing. Now, right now, there may be discussions going on between, research hospitals that are saying, hey, you know what, we are seeing a whole bunch more clots. Perhaps we should be looking into this. Perhaps, but for all we know, there's probably a big multi-center grant and grant proposal submitted saying we need $50 million over 10 years to look at and understand this phenomenon. Perhaps they are looking at it. I don't know. We don't know. Right, you're just always going to be working with a whole degree of unknowns as the public has to try to sort of work in real time with the events happening as they do. And what are we? And why are we? What are those events? It's people dying unexpectedly. There was a study out in JAMA last week, so it might have been the week before, where they're just admitting, cardiac events in marathons have shot up, whatever, a significant percentage since 2020, and particularly, I want to say 2023 was a sort of standout year, and that's probably, we don't know the data for 2024 and 2025 yet, because we don't know if they're still probably doing the analysis, okay? But the data is there.
Dire enough that people who work in these places understand that it affects all of us. And like even, I know the ABS is no longer publishing excess deaths, the actual numbers, they're doing it as an estimate. So a lot of this data that we don't get to see, no one's getting to see it.
Right.
The UK. How can we find the science of what's actually going on if we don't have the data?
Right. And the UK, so if I shoot up my slides. So there was, what was he called? See if I can find it. There was a guy on Twitter who was doing UK deaths, right? This is it. What was he called? Outside Alan on Twitter. And what are we looking at here? You're looking at all-cause mortality. So this is weeks ending 27th of March 20 to 27th of October 23. Okay, and where it's red and black, you're seeing extreme increases in all-cause mortality. Now, the disturbing thing to look at is that as you work down, you're working across the years of the mass casualty biowarfare event, and we see, and if you look here, so this is the age categories across the top: 0 to 24, 25 to 49, 50, and older you get, right, is the as you work left to right. Now, at the outbreak or the start, it generally hit the older population and generally sort of stayed like that. You're still seeing some in the first year going into 2020, 2021, right? So this is not vaccine related. Now, some will argue that this is entirely hospital protocols. Maybe. But I'm not so, I wouldn't speak so confidently as to the source of these deaths, right? Especially when it's happening in the young, and you are dealing with a novel pathogen that we do know has this coagulopathy component to it. Now, for sure, were they taking people because their blood oxygen saturations were dropping so precipitously, and were they putting them on ventilation. Yeah, they were. But that doesn't mean they would have got out of the disease state that they were in had they not had ventilation done to them. Okay. As you proceed through the years, high all-cause mortality. So on the right hand here, as we look at the latest data that we had available to us, it goes back to normal levels. Where you keep seeing excess death is in the younger cohorts, in the 25 to sort of 50-year-old cohorts. And that's your working population, that's your parenting population, that's an assault on the carrying capacity of any country and society. And you can argue the cows come home, what's the source of that? I'm not going to do that right now. All I'm going to say is I know that there's because I'm looking at it, right? With this instrumentation, I'm looking at misfolded fibrin. There's the cause for the, what I would say accounts for the majority of the disease burden that we're seeing, okay? And how that maps out, etc., is a complex thing, and it's not the topic of discussion for today. Okay, so where did I?
That is a very interesting thing. I actually haven't seen that before. As to why I'd say the older population isn't matching that black-red.
Yeah, it's disturbing. And what did the UK do? In, I want to say, at the turnover of 23 into 24, they basically changed their whole formulation for reporting on deaths. And so you can't do that when there's a obvious signal in your data that and you could perhaps understand it if it was the elder end of the population, but when you're seeing such heavy impact in the younger generation, and you don't go changing how you record and report deaths to the public, but they went ahead and did it. And that means, under the current circumstances, I'm inclined to be less confident in the output of the work that they're doing. Okay. Now, I don't know.
Oh, well, they're not the only ones, so it seems to be they've passed that message around, that memo.
Yeah. So that's not the one. This is an older presentation. So let's go. This one. I've got so many presentations open that I'm trying to find Raman spectroscopy. That's not it. What wait a second, just because I think this is important.
It's okay, we can edit it out if it takes
Right.
a lot of time to find it.
Okay, so using I'm annoyed because, well, okay, so actually this slide, if I just copy this, so I've had to expend a lot of effort into you want to be able to categorize the molecular structures that you're seeing, and in order to do that, one of the more cutting edge ways to do that is through something called Raman spectroscopy. Okay, Now, what you're looking at, see this little box here I'm moving around,
Yeah.
right? So as we're beginning to learn about the fundamental nature and structure of amyloids, we can begin to say, what do they have in common across different proteinaceous species? Okay. And what do we have here? So we have alpha-synuclein at the bottom in light blue, amyloid beta, apolipoprotein, RPT, I'm not sure about, and HET, I would have to go and read the manuscript. But the key takeaway here is that at around 1700 centimeters, which is just essentially you can just think of it as a arbitrary, it's not arbitrary, but it's a measurement of vibrational activity under something called Raman spectroscopy, where there is a increase in signal relative to noise in the background. And at a particular wavelength, you can say this corresponds to this structure if you were looking through other more canonical methods like Western blotting or other mass spec type analyses. In fact, mass spec analyses are not as good at looking for this amyloid stacking as the Raman spectroscopy is. Raman spectroscopy is in fact turning out to be one of the best methods that we have. Problem is that a Raman scope will cost you $1 million and very, very few people have access to it. I'm lucky I have access to it. It's because of that scope I was able to reject the nonsense coming from the graphene nanobot side of the internet. And what do I see when I take the slide, literally the slides that we were looking at earlier, right, which I've just shown you through the light microscopy, okay, down through the electron microscopy. If I do Raman spectroscopy, what you can see is in this is you get a peak around 1700 centimeters, which says, yes, there's an amyloidogenic core in that peptide that we're looking at. Okay, so now we've ticked off another one of those boxes, and it's one of those boxes which is at the bleeding edge of diagnostics with respect to understanding the molecular properties of any protein that you wish to be looking at. All right, now that then brings us to this, which is RT-QuIC. So RT-QuIC stands for real-time quaking induced conversion. Again, I explained earlier in the interview, you take a well plate, something like this. It's got 96 little wells in it. You see that?
Yep.
Right, and you fill them up with different combinations of peptides and whatnot. And the idea with RT-QuIC is that you take, and it's primarily used for looking for Creutzfeldt-Jakob, chronic wasting disease. You're seeing an acceptance for, I want to say it's not, is it alpha-synuclein? Well, all the sort of peptides that you could generally get from a spinal tap that could point to there being the onset of neurodegeneration, even though it's not visible. The person is still in the what's called prodromal phase. Okay, you might see hints of it, but the full disease expression isn't there yet. And you can take you can do a puncture of the spine, take the CSF, run it through RT-QuIC, and then see if you can cause the reaction where you take a seed amount of the peptide and then you run it against the so you take your sample from CSF, you have a normal bulk solution of the peptide, and you see, does it does it cause the amalgamation into beta-pleated sheets such that it causes an increase of fluorescence per cycle that you run. And this runs for, you can do anything from like 24 hours to a week in trying to sort of investigate this phenomenon. And I've spent like the last year essentially trying to use recombinant peptides just to see if that prion signal is there that was reported by Hammerstrom and Nystrom. And I can point to that work here, hopefully. Get this, right? That's Hammerstrom and Nystrom's work. And just this little cluster down here, spike 532 seed, specifically causes the aggregation of prion protein into the scrapie form. I can say I was unable to really replicate that. Some signals, but not really very strong. When I took a seed amount of the misfolded fibrin, so this is me running like all those little squares. That's basically a representation of all the wells that I've got running under reaction for the RT-QuIC. And when I was doing this, I was like, I'll just test quickly the clot against plasma. So I draw blood, I separate out the plasma, I put it into a well, and after a year of using or trying to get a signal, I get a massive signal with RT-QuIC with this seed amount of calamari versus my plasma, right? Which is what these two boxes or two bars here are indicating. Everything else, all the reactions I'm running, there's whole combinations of different peptides, recombinant peptides running, and there's nothing happening. It's all within sort of control range. The two wells in which I had enough plasma and enough plasma to fill those wells and then put in a small seed amount of the calamari clot. Yeah, I got a huge signal.
Were you surprised?
Was I surprised?
After, a year of trying with spike?
Well, yeah, it was nice. Well, let's say, when you're building out a lab, especially when you're sort of starting from scratch and you're working under the conditions that I am, which is you are like every penny counts because you've got to be very strategic in how you deploy your resources. You want to be, you want to know that you're capable of capturing the phenomenon that you're looking for, right? And, after a year of trying with recombinant peptides and, primarily around spike protein, prion protein, et cetera, I wasn't seeing anything. And yeah, there is this sort of anxiety that a scientist is going to feel that you're messing up somewhere in your protocol such that it's your fault rather than the principles behind the study that you're doing. Okay. And so to see a signal there, I'm like, I'm not doing it wrong, right? I'm concerned about the validity of trying to use recombinant peptide. I think not the validity. You just need way more infrastructure, and I think there's just way more steps you need to do to be sure that the peptide is functional when you receive it in its lyophilized form. Okay, and primarily those peptides are used for assay, right? So, when you do like a Western blot, if you've got a sample, so you take a sample from someone's spinal fluid, you want to look for what peptides are in there. So you ask the recombinant peptide company, make me this peptide so you can map out its molecular weight with a dye. And then you see the expression profile that you get from your patient, and you see, does it match what you think might be the abnormal peptide in this instance? And I'm hurling at you a whole bunch of complexities with respect to the how these systems work and interrelate to each other. But I'm hoping that just people will go back, listen to this multiple times, and try to absorb what it is that's being said, because you need to learn this stuff, right? And you must get on top of it for what it means. So to see this signal is not surprising, but it's not surprising considering what we know when we've looked at the scanning electron microscopy, we've looked at the ultraviolet histology with respect to using fireflavin and light microscopy, the signal that we've seen with Raman spectroscopy. This is just sort of like the final, horrible euphemism, but in this case, the final nail in the coffin, right?
Yeah.
And so that suddenly, that signal then opens up a whole bunch of potentialities that I'm concerned about. And so immediately, right, this, and I only got that data a few weeks ago, right? And so I'm trying to, like, so let's try and parse out what this means with respect to the public at large. So first off, I'm fairly confident that most blood products post-COVID are likely contaminated with probably seed amounts of misfolded fibrin. Okay. Now, you know what, that was a question I was going to ask you because I was talking about it the other day, which was, there was a case in New Zealand.
Yeah.
And
I'm pretty sure that we've heard that, you know, people have died from
right. So the baby where the state stepped in, that right, there was a case where the parents were not wanting blood transfusions from the sort of public supply of
yeah, I think in that particular case, because it was so high profile, I don't think the baby passed away. It wasn't that case, but I from my memory of that, it was that family had read of another case or had been notified that this had been happening.
You see, so I'd like to clarify, right? So if anyone has the reports or the tweets, if they haven't been nuked off the internet, but I'm pretty sure that they did do the surgical procedure, and there were post-surgical complications of clotting.
Oh, okay.
Right. That's what I remember. Now, did the baby pass away? I'm not sure. I don't think so. But yes, there were problems, if I remember correctly. But you know, memory being what it is, you know, I'd like to see it in sort of black and white. But anyway.
I mean, we do have a precedent for all of this in the whole infected blood scandal that's
Yeah.
out in the UK anyway. So it's not a stretch to imagine that if, you know, there are these seeded peptides or something, if the blood is contaminated with anything, literally, that it's going to cause issues if they're not doing any screening for it. So
and look,
obviously it's very dire, but you know, we do have a precedent which went on for decades where they covered that up, and it's only recently in the UK where they've sort of admitted to it and given some financial compensation. In Australia, they're still not doing that.
Yeah, and that's the machine, man. It always is, and the people expecting some sort of snap of the fingers and everything to right itself at the moment. No, this is, you're a long way away from that, and because of the scope of what it is that we're dealing with, anyone in any position where they're going to be held accountable is going to do anything and everything to survive to the point where they can pull their pension and get out of Dodge, right? So, you know, the concern here, right, is suddenly there looks like there's an infective peptide. If this sample that I've been running has been out of the body for 3 months, even though it's been out of the body 3 months, once it comes into contact with fresh plasma, is able to initiate the misfolding and aggregation into a amyloidogenic form, such that you're seeing an increase in fluorescence. This is a big concern, and it's one of the sort of primary measures around how you would define scrapey prion. right? So you can take scrapie prion and you can take femtogram amounts, and it can be infective years after, right? So the operational principle around which they work with prions is there is no safe exposure dose, right? And, you know, when they do the experiments, you know, it's literally one, in terms of dilution, you can basically say it's one molecule or one piece of misfolded peptide is enough to initiate the chain reaction under experimental conditions, such that essentially it's a lethal pathway in terms of experiment. Okay, now how much that relates to everyday exposures, you know, not everyone died from BSE, right? Now, what's the injury rate after that? I'm not sure we really know. You know, again, misfolding peptides manifest disease in many different ways, and there may be all manner of fallout from the mad cow outbreak that we just we just put it down to it being part of the background level of disease burden. Okay?
We'll never know, really, will we? I mean,
no. I mean, there was a study done over many years where they were using antibodies, and this was before really these amplification methods were available, and they were doing antibody staining looking at people's appendix, and I can never remember if it was 1 in 300 or 1 in 3,000. I'm gonna go, let me just because that's an important number to know. ChatGPT will know. Examining index. What was the population penetrance of CJD in the UK? Let's just see what so sample 8,318 appendices and estimated prevalence 3.7 per million. So 1 in 2,000 people is carrying the scrapie prion. And that's a coarse measurement, trying to do it with a microscope and just seeing if you got a positive hit with antibody staining. I would imagine if they took RT-QuIC to those tissues, they would probably see a much larger number. But in this instance, they've got a number. The government can say, "Oh, we've done the study." Boom! It gets put into the libraries, and they've done their bit, right? And they just hope they don't get called out on it. So, with getting back to what we're looking at here, so we're seeing something that looks infective. It's been out of the body, and this would fit the canonical description of prion-like disorders, and that's a huge concern. So I've been working feverishly to try to see if I can figure out what's been going on. And so, you know, I had to wait to get this, which is basically just an ultrasonic disintegrator of tissue and what have you, just to be able to sort of break it down into a fine form where, you know, I was generally having to do it sort of manually, and in this way I can get a finer titration of the amount going into per well of peptide, even though I'm saying before when I had basically a coarse non-ultrasonicated preparation, I'm unsure of the amount of seed material that's going in, just because of the limitations of the methods available in the laboratory. So in this instance, now you can see that there's like a uniform distribution in the beaker, right? And in this instance, this is I've basically just diluted it 50 times, okay? And if that was scrapie prion, that would still, that concentration light up all the tests. And in this instance, so you're just looking at these sort of black squares, or sorry, black lines. Each one is a well. This is the control material. And I'm not seeing a response, okay? And that's, I hope, is a good sign that there is, we're not looking at something as severe as mass exposure to scrapie prion. But I still think there should be concern about what is or what had infective properties months after coming out of a cadaver, right? Let alone what's considered live blood and put into the blood supply for distribution to hospitals for, you know, whatever treatment, whether it's emergency or, you know, people who've got cancer that need blood transfusions, et cetera, et cetera. That's, you know.
Because that's not diluted. So I'm guessing you're saying is that, like, in terms of everyday exposure, you're thinking that it's you know, to say surfaces or saliva, maybe, obviously, and we can get.
Well, do I think there's a risk from shedding to this peptide? Yes, I do.
Okay.
But is it at the level of concern where I would be talking, where you're talking about chronic wasting disease, scrapie, BSE, et cetera, et cetera? I'm still, I can't give a definitive answer because I'm literally just working through the tests at the moment. But
And so that's what we're looking at in terms of getting you funded and continuing your funding, is to assess the disease risk from what you've recently discovered.
yeah, and all I can do is just iterate through the peptides and the well, there's just many there's many different factors that need to come together, and I'm dependent again. Like, I don't go to the big money players and ask them for money because that comes always with strings attached, right? I would rather do this and ask to people. You know, what I do at the moment is I basically say, I need this bit of equipment in the laboratory, right? And, you know, you don't have to send me money, send me equipment, right? Just so you're sure that I'm not I'm not running away with it or doing anything untoward. I'm working literally every day with this, at this problem, right? And I have been for essentially the last 5 years.
I mean, I think the
And
question people always commonly ask, and this is sort of part of the cognitive dissonance, is, well, how do they stay safe? Or how do they heal from, say, something like long COVID, issues with shedding?
okay, so one of the things that I found I can do is that let's see, let's go back up through this. No, maybe the other way. So there are two issues that you're dealing with. One is those who have been injured, and those who have long-term sequelae from the infection itself. Both exist, okay, and are often struggling with significant disease burden and are often being gaslit by medical professionals in that their symptoms are dismissed or they're told that they're psychosomatic. And what I've found is that, you know, if we understand this clotting disorder to be a sort of universal key to understanding the symptoms, then it's possible to be able to test individuals and just get an idea of their amyloid microclot burden. Okay. Now, there's no automated test to do this, and it's not on the tick box of standardized tests that your GP or clinician has available to them. And 9 times out of 10, most people go to a GP and you get 5 minutes and pushed out the door as quickly as possible as they try to work through their caseload for the day, right? And you going in and asking for specialized tests that are only available in very, very few places just often doesn't get you very far. Right? But what we can do is we can look at, so this is someone who is injured from Moderna, okay? And here, what you're looking at is this is what I see all the time now. It's a microclot fibrillar form, and I tend to see that, well, how to classify them. Punctate, generally circular type microclots, more, how would you say? Almost like sort of aggregations of sort of like tissue, if you like, sort of tore up tissue paper that's sort of stuck together. And then there is this fibrillar form that often has a sort of rotational element to it. And we've spent a lot of time talking about this fibrillar form or this twisting of the peptide as being at the heart of the pathology that we're dealing with, because that's the proteinopathy expressing itself, right, as a misfolded peptide. Okay, so you take that, you process it, so you can check it with ultraviolet, and then I can discern whether those objects that you can see under light microscopy are there, and start ticking the criteria for amyloid-like deposits, okay, or amyloid-like inclusions in the blood. And this, you know, it's unfortunately, again, it's not quantitative. It's not quantitative like RT-QuIC. Now, potentially, if someone was going to fund me, I could try to do RT-QuIC off people's blood if they're if they have or long COVID, and see if it's triggering, and you get an amplification out of it with respect to fibrin. That's an approach to take. But in this instance, I can take people's blood, I can process it histologically, I can look at it under a microscope, and I can tell you, I can give you an assessment of how much amyloid you're carrying per sample of blood, okay? And so, so there's, in a sense, there's a degree of sort of hope here that a lot of the battle is people just trying to get the medical system to recognize that they have a problem. And there are even medications that you could be put on, right? There are antifibrinotics that you can take should you have a problem with, you know, they think that clotting is one of your issues. You know, these pathologies existed before. It's just we've got another way of inducing them that's been introduced to the environment. Okay, and you know, I know some people are, certainly with some justification now, suspicious just of the pharma medical complex. And you know, there are multiple ways to skin a cat in this instance and try to get symptomatic relief. And you know, like I say, I can't risk my scientific objectivity by saying go and use X product. I can tell you, collaborative groups that I'm using, I'm working with, are seeing very, very good results through their approach. And you know, if people want to,
I mean, in theory, you could do this blood test do whatever things that you're going to do, whether that's those anti-fibro medicine or natural things, then send another blood test.
Yeah.
You,
Yeah.
and then compare what's
Yeah.
effectively working and what's not working.
Yep. And that's what I've been doing. And you know, that's going a long way to sort of keeping the lights on at the moment. It's just me assessing people's blood. Now, you know, it's not a full objective scientific research project, right? Because the people who are sending me samples are people who literally have issues, right? So that's always going to skew your sample cohort, and so it's difficult to say again what's the penetrance into the population. But I'm pretty certain at the moment that it's probably higher than they're letting on right now. And again, the problem, because the people that write to me are saying, yeah, I have this, I tried to go to the doctor and I'm basically just brushed off, right? Or I'm given you know, it's incredibly frustrating, because what happens is that people get labeled with what's called functional neurological disorder, which basically means they don't know the cause and basically are trying to say to you it's essentially psychosomatic. And in this instance, with this data, you're able to push back against that diagnosis, right? It's not a psychiatric illness. There can be an underlying pathology, and then if there's
tests, right? The medical system, they love tests. You get a test done, you hand it to your doctor, and you ask them to explain it.
Yeah.
But
they don't offer this test, right? I can do it, right? So, like I say, if you want to help and you want to keep this research going, and you have a problem, you want me to look at it, I will. And you know, usually the price to do that is a minimum $300. I won't even charge you unless you want me to actually give you a report back, right? I can look at it, and you know, if you just want to add to the library of samples that I have, I appreciate that, right? Because it helps me build a better picture clinically of what's going on. But yeah, if you've been, I was abused it's probably not the correct way of thinking, but it's just you're looking at a system with limited resources, limited time, and they have to deal with hundreds of thousands of individuals on a yearly basis. They have to find a way to sort of categorize you and give you some treatment, right? It's just that in this instance, labeling people as having functional neurological disorder is the incorrect diagnosis, when we potentially can look for pathological signatures and say, look, there's something we need to be treating, and there are methods to at least try to mitigate amyloid burden, especially if it's in an easier to reach compartment of the body as the blood.
Did you want to bring up your website, Synaptic Labs?
Yeah, why not? That's always useful. Yeah, so you can go here, Synaptic Labs protocol on sending blood samples. If you go down through that, there's a little video, 3-4 minute video of me just showing what you need to do, and what I will do is perhaps let me just play this, because then I can grab a drink, and then we can sit there, and we can just sort of decompress for a little bit, and then if you've got any questions after this, and then we can wrap up. Is that okay?
Yep, sure.
Sure. Okay. So, and I'm not going to listen to. I hate listening to my voice, but just tell me if you hear the sound. This is the suggested protocol for administering. I'm going to get a drink. A needle stick to get a blood sample to send for amyloid burden testing by UV microscopy. First, what are the items that you will need? I suggest alcohol, sterilizing alcohol and swabs. These items are given in the list associated with this clip. And although there are differences, slight differences here with respect to methodology, I'll be using a syringe needle rather than a needle stick device that is commonly used for diabetes. But the primary purpose is to show you how to take the blood sample, apply it to the slides in preparation for posting to Japan. So I have two microscope slides that I have ready to put the blood samples on. I'm going to sterilize the finger with alcohol. Never easy, but we just need a few drops of blood. So here goes. So that's the needle stick made. I'll withdraw, and hopefully, if we're successful, you can see that we have got some blood. Let's hope we can get enough. Yep, if you just squeeze it. Now I'm going to apply to the microscope slide. 1, 2, see if we can get some more. 3, 4. What I do suggest is try to even out the samples that you've put. A method to do that is just use another glass slide and just spread the blood out so it's a thinner layer, and you want to be aiming for around 2 by 2 centimeters of coverage each one of these slides. Slightly larger than I would have wanted. And that there is the would be ideal for analysis. Let these dry overnight in a dust-free environment and follow the rest of the instructions included in this methodology. Thank you for all those that are contributing to this work. As my voice stopped, I'll just so if below that is links to where you can get, so I'm just looking at Amazon.com here. This is and this is the bottom range of cost for microscope slides, and you also, there's a link to, I used a hypodermic needle just because that's what I had laying around, but I would encourage people to just get the little lancets that diabetics use. Again, $7 off Amazon. And sterilizing swab alcohol, everyone knows what that is. I would encourage people to look for, so link, these types of casings if you can, right? So you can basically get 2 microscope slides in there. Best to send two because I have had cases where they've broken one, even with protection and what have you, just to sort of increase chances of being able to get a useful sample from. And with that, I can give you, well, the person, some idea as to this microclotting burden. And yeah, again, fortunately, there aren't automated ways to do it, and there isn't a tick box that your GP can tick for standard blood tests. Unfortunately, you might try for a D-dimer test, maybe, but I think the problem with that is that you might have elevated D-dimer after initial exposure, as the clotting is sort of initiated generally, just because you've got systemic coagulopathy from spike protein. But once that sort of signal has died down, the body, because of it's not going to signal that there's sort of like a clotting emergency with misfolded fibrin, right? And that fibrin is just likely to just continue to aggregate and aggregate till we end up with those proteinaceous rubber-like clots that we were looking at earlier. If people want to spend a bit extra for the lab, I like this brand. It's called, it's Japanese brand, Matsunami. Very clean slides. The cleaner the slide, the better for me, because there's just less confounding objects. But if people want a link to where I get those slides, and it's approximately about $50 for, that is 100 slides maybe. Yeah, 100 slides. And if people want, you can get a smaller vacutainer than this, 5 ml of blood. And you might have more problems sending that via the post. If you're just doing the glass slides, there's no problem. You can send that in standard post and just put dried blood sample microscope slide. That gets through, no problem. And a few times a week I get a sample delivered, and I've got a backlog at the moment just because the scope I had was out of commission for a month or two. And yeah, like I say, if you think you've been hurt either through the or from COVID, here's one way that you can get a diagnostic, especially, like I say, and I know because I've been through it years ago when I got hurt via post-viral syndrome. It was very debilitating for many years. I don't know. Well, I still think I have to struggle with it these days, but yeah. And people can reach me through that page. The address is all there, contacts, and if you have questions prior to trying to make the sample, you can email me. I say, like the Discord is it's a rowdy bar type of environment, just think of it like that. But if you can put up with that, or you can just ignore it and you want to reach me, Discord is a good way to do that as well. Twitter, not so much, just because there's so much spam DMs now as a consequence, so it's difficult. Basically, I'm just deleting DMs in Twitter just because it's just all looks like nonsense now. So yeah, I don't know if you had any further questions.
Can you close that screen down, Kevin?
I can.
Yeah. Oh, look, thank you so much for all of this. I think we've covered a lot in the time. Did you ever test your own? I mean, it's just curious. Did you test your own blood in it?
Yeah.
Have you found?
No, mine's pretty clear, but then, I have access to the countermeasures that are being deployed on sort of clinical testing elsewhere, right? And you know, well, I can show you the product, right? And
Yeah,
I'll just
you may as well. No one else, everyone else
no,
is doing it. You're not making a buck from it.
I'm not selling it, right? It's
Yeah,
got
yeah.
nothing to, right? I
I'm
don't get it.
curious. I'm curious because if I had access to that machine, you'd be testing, you know, those people closest to you.
Let's say I'm more than happy to put you in touch with the people that are making this, and you can speak to them about their data. And but well, the version I have, they sell it. It's called medicinals. The one I have is just labeled for a different market. It's called Molecusan in this instance, and it's very, very high grade mix of active phytochemicals in this instance, and basically combination therapies to one, induce, so sort of senolytics to take out infected cells that are potentially sort of zombie-like cells that are just spitting out peptides, binders so that the spike protein that is released is captured and excreted, and targeted molecules to, again, it's sort of trigger sort of apoptotic pathways and deal with circulating. So one of the targets is primary blood monocytes of the immune system. So what they find is that there's a high spike burden in people with who are suffering sort of sequelae either from vaccination or infection. And they find that if they, by targeting those and exosomes, where they'll find, in extreme cases, they can find 1,000 picograms of spike protein on average per monocyte. They can reduce that down to essentially negligible values, sort of in the tens of picograms. And in severe cases, one of the clinical approaches that's being taken is to do heparinized apheresis, which is essentially taking the blood, essentially like you would if you were sort of donating blood.
Yeah.
The blood is circulated through a whole bunch of filters. There are different types. So heparinized apheresis is harder to get. There are other machines which are more available, less sophisticated, called EBOO machines, and they're getting good results with those, but the gold standard is the sort of heparinized apheresis, and that coupled with sort of multi-month treatment with these products, they're recovering many patients. I would say probably way more than the orthodox health systems. And so I don't get any money from selling
that's
that.
really good news, really, because I think what we've sort of established today is that if there is testing available, like you can obviously rule out if it's amyloids or not by getting your blood tested, which will steer you in a certain direction. And if you are taking any kind of treatment, you will be able to also assess ongoing by doing another test gathering whether it's working. And of course, for scientific research, for all of us, we'll be able to see what's working and what's not working.
Yeah, and
there's a lot of stuff out there right now is just like, take this on blind faith, and if you're feeling better, or even using medical tests aren't necessarily sufficient because they're not testing amyloid burden.
No.
And so you might even pass a medical test, but it's not actually.
Yeah, you're still feeling terrible, right? You can't get, people can't get through a working day. And again, the end diagnostic that they'll give these people, FND, functional neurological disorder, and probably slap you on some SSRIs and mood balancers and antipsychotics. That will be their approach, and it's when we have tools available to us that can prevent that sort of misdiagnosis. I would encourage people to sort of at least take it and try it for a few months, and I'm pretty confident we can tell the difference pre and post treatment. A lot of people, I say a lot, but I have got people that have contacted me who are already on treatment that just said, can you please tell me how it looks? And that's it provides a sort of peace of mind for people, and just not just peace of mind. You want something that nearly, I would say 90% of the people that have sent me blood, when I've written back to them with their results and said, look, here you go, you've got this, I'll send you a full report on all the images that I take, and often I'll send people I'll video record myself working through the slide and just giving my commentary, send that back as well. And that gives people hope and something to go to their health practitioner with and say, this is where we need to be working. And with that, you can start working towards trying to regain your health. Are you going to get back to normal? I don't know. I'm not saying that. I'm not a doctor in this instance, and I work strictly on the research side, and and I say I'm not making anything from this. Don't sell it. Don't do. There's no product page on my websites. It's purely if people want to contribute because they think this work's important, that's available. And if they want to test and they want to send a couple of slides with your email and with your medical history, please just put in a printed page or handwritten page of the basics, age, past medical history, what you're going through right now, current treatments, and I can, and it's working pretty well.
And ultimately, that's the end goal, because we may never know the actual, weapon system in its.
Well, I think we have a pretty good idea now. Actually, yeah. I'm confident now, right? Whereas before, there was always that little bit of ambiguity because they weren't admitting to it. Whereas literally, oh yeah, they've just put their hands up now and just said, yeah, okay, so, yeah, there were ill military.
I guess we're winning in a way. I mean, when you think about it, where we
were,
I mean, it's taken a long slog, but like the pendulum has definitely shifted to the bioweapons angle.
Yeah, but they've got away with it. They've got away with it because the only people that left are those that have been impacted directly, whereas the population where they haven't lost loved ones, they had no adverse events. They've got their lives to get on with, and so whereas the whole world was keyed in at the beginning just because of the novelty factor, and yeah, there were people dying. Again, people are going to argue about the source of the deaths, but there's certainly a increased disease burden out there that can't be argued with. They managed, right? They really managed to kick that can far enough down the road that you're not ever going to see the mobs of pitchforks and torches bearing down.
So are you still feeling like we're sitting on a ticking time bomb, or are you feeling like there's a lot more hope now, like since from this time last year?
Good question. And look, I'm working as fast and as hard as I can to try to understand the impact of what that misfolded fibrin is. And what I can say is, I've seen it be infective post-mortem. I would suspect it to be infective via potential exposure routes. And the problem is, primarily, a lot of your stool contains broken down fibrin, right? As just the liver doing its thing. And so I think there is an environmental seeding, and I would say it's with high probability that blood products used on a day-to-day basis are likely contaminated at this point.
And what about, I know there's been lots of talk on social media, sexual contact.
I don't know. I hope not. But there's still the disturbing trend in drops in birth that is ongoing in many countries, and the argument is, it's kind of heading that way anyway. But I would say generally, look, if you're in a sort of settled relationship, family, etc., it doesn't matter. You're passing backwards and forwards anyway. So, but if you're dipping into the dating pool, probably suggest, discreetly ask about their medical history. I don't know, just the usual.
What a world we're living in today, isn't it? I mean, it's quite crazy. Is there anything else you wanted to add? I think I've pretty much exhausted. I think we've done a really good job today of putting it all together.
Yeah, it's quite long.
So I
don't know.
If anyone wants to watch it all from start to finish, and they will get the big picture, go back to the last video and get the kind of background around prions and so forth. It'll tie together. Is there anything you wanted to add, Kevin? Otherwise I'll just close.
No, I think that's it, Jeannie. I just hope the message gets out there. And like I say, between this time and last year, like I've managed to work out at least to give people some idea as to this, like the more complex proteinopathies, let me worry about that right now, right? What we can see is that there's a blood-borne one, right? There's one in the blood. It's a kind of low-hanging fruit in terms of trying to address people's symptomatic profiles, and there's a way to at least get a handle on that, and I can point you to the people who are making effective interventions at the moment.
That's great. That's great to know. I say, in closing, Health Alliance Australia opposes harmful health measures, laws and mandates, provides educational content. Please feel free to share this interview and information widely, cut, copy, paste, distribute. It's important and heavily censored. Get it out there. When sharing, please do link back to Dr. Kevin McCairn's website, McCairn Dojo, and his Synaptic Labs website, healthallianceaustralia.org if you can. I'll be putting all the links plus a report which includes the slides, important information, links that we've discussed here today, and also Kevin's report on that page. So it's all in one place. You can visit healthallianceaustralia.org to join our mailing list, become a supporter, or donate. Health Alliance Australia, myself and Kevin, Dr. Kevin McCairn are also on LinkedIn. You can join us there as well. Thank you, Dr. Kevin McCairn, for your time today. And thanks everyone for watching.
You're welcome, Jeannie. Thank you.